What we're trying to do today, and in the several talks, would be the developments in Parkinson's disease, and also later on you'll hear about clinical trials and how drugs are developed. And it's again very, very important because one of the things that I'm also involved in the research and development stream of the Department of Health in the UK. And we were in a meeting on Monday, and one of the things that was discussed at length is why is it in the UK we have difficulty getting people with Parkinson's involved in clinical research. And a policy in my hospital, my unit, is that every patient who comes to our clinic is given a little brochure which says that we would be asking you to join or take part in clinical trials and research unless you explicitly say no. And more recently, we've got an expert patient group who have formed a little video. And this, again, we will hear from Richard Windle later. But again, in that video, they've spoken about the importance of taking part in trials, because really that's how new developments will occur. So we are totally dependent on your participation for this. The first question is, why do we need new therapies in Parkinson's? And I think you would all know that the key hormone or protein or peptide in the brain that's deficient in Parkinson's is dopamine. We also know there are, of course, other chemicals involved as well, serotonin, acetylcholine, etc. But dopamine by far is probably the most important one. It sends messages from the brain, parts of the brain, to coordinate movements. So essentially the basal ganglia, which is the coordinating center within the brain for dopamine, will send messages to all the motor area, will allow me to do all these hand movements, for instance, I'm doing. So overdrive of dopamine can cause problems, and of course dopamine deficiency will lead to many of the symptoms which you would experience, such as slowness, stiffness, difficulty in turning in bed at night, etc., etc. And essentially, for reasons that are unclear still, in Parkinson's we have loss of this dopamine cells, which are faster than what we lose with normal aging process. So if we all lived up to 150, we'll develop signs of Parkinson's. But for people who develop in lifetime, it's usually accelerated loss of those cells during lifetime. And this is basically the loss of the cells are important, and we now know that when we are picking up people with Parkinson's, when your diagnosis is being made in the clinic or in the GP surgery, about 70% cells have already been lost. So another key challenge for us in research is to really pick the condition up way before the motor problems start. And we know, for instance, loss of sense of smell could be a very early indicator in the development of Parkinson's now. And very recently I've just come back from a meeting where it was suggested that this can occur anything up to 10 to 20 years before the stiffness and the tremors start occurring. So the challenge for us is pick up this early period, but it's worth noting that by the time that the tremor and the slowness starts, we've already got about 70% cell loss in those dopamine cells. And in terms of therefore new treatments, one of the things would be that to think how can we replace this dopamine, and one of the ways is to produce chemicals in the brain that will mimic the effect of dopamine. Fairly simple, as for instance, analogy will be with diabetes, where you're trying to mimic the effect of insulin, for instance, and also preventing the breakdown of the cells. So there are two approaches. One is increasing or maintaining the formation of the chemical. Second is preventing the breakdown of the different cells, dopamine cells that are dying in Parkinson's. And the reason why I think we need therapies, the new therapies, are that the limitations of the current therapies. Now again, you would probably be familiar with a lot of this. Dopamine agonists, many of you will be familiar with them, pramipexole, ropinirole, rotigotine patch, and so on. and levodopa preparations, Sinemet, Madopa, Stalevo, are all very effective, but the greatest beneficial effect of these drugs will probably manifest in what we would think is the early stage of Parkinson's. And in essence, that's the first variable a little bit, but between 5 to 10 years of Parkinson's. But as the condition progresses, two problems emerge. Firstly, of course, we don't have a cure for Parkinson's with any of the current therapies that are available to us. Neither do we have a really robust treatment that will slow down or stop the progression of the condition. Now, we have that for multiple sclerosis now. We have treatment which we know affects the progression of Parkinson's. So if one had a treatment that can actually maybe not cure the condition, but slow down the way the degeneration of the cells, even that would be very, very useful, but we don't really have that at the moment. And also, as the condition progresses, that's the other aspect of it, the drugs become less effective. So, you know, when you have a condition, your Sinemet plus 3 times a day was very effective, but then people start getting wearing off, getting dyskinesias, and you need to take more and more of the drugs. And that's because one side there's progression of the condition, other side there's less efficacy of the drug as well. So all of these factors would suggest why we need new treatments or new strategies, which will at least try and address some of these concerns. Let's now talk about 3 new strategies that we might be looking at. Firstly is drug therapies that we call protein therapy, so they would utilize proteins. And these are actually proteins that are usually found within the brain. And the generic name for this is growth factors, and there are many of them. There's brain-derived growth factors, there's glial-derived growth factors, etc. And there is evidence in animal model work that if you expose cells which are exposed to the toxic effect which might be causing cell death, then these proteins will actually protect against the cell death. So therein brings in the protective part of it. And secondly, they'll also often reverse the cell death as well. Now all of that looks very promising indeed in terms of therapy. Another area we have to remember is that if you're using these sort of proteins for treatment, if you give it by general route, let's say by oral route and so on, we have a problem with the blood-brain barrier. Now, for people who might not be familiar with it, that there is a barrier between the circulatory bloodstream and what gets into the brain. As a result, for instance, when you take your levodopa tablet, only about 10% would get into the brain, regardless of how much you're taking into the stomach. And that's partly because the blood-brain barrier is a specialist layer of cells, and that stops the materials. It's a protective mechanism of our human body to protect the brain, because our circulation is open to all sorts of toxin ingestion, for instance. And again, as I said, oral, something you take by mouth or inject, cannot reach their site of action in the brain altogether. So this is very important to remember in terms of delivery of the protein. So ideally, if you want an agent to exert its full effect on the brain, because they have to enter the brain to be effective, you would probably want a procedure that can actually deliver the product straight into the brain. And proteins, of course, can reach the brain in different ways, but direct infusion is probably the most important way, and I think Alex is the expert in this field who would probably tell you either through catheters or through pumps. And this has been used for many, many other conditions where protein infusion has been performed to the brain. And one example about a protein is a drug called GDNF. Now you might be again familiar with this, Glial Derived neurotrophic factor. Now glial cells are very important in Parkinson's. They are cells which surround the dopamine cells. They are important for the survival of the cells, and we now know that glial cells have inflammation. Something happens in Parkinson's that these cells are inflamed, and neurotrophic factor can actually reduce that amount of inflammation. And in other words, these are a group of proteins we call trophic factors. Remember, factors, neurotrophic factors of proteins. They're important for survival of the dopamine nerve cells in the brain. And these can also make nerve endings grow. So not only are they surviving, but they're helping growth of new cells. And also, if you look at animal models, so people have been working on various animal model projects here in Oxford, and for instance, at King's with Peter Jenner's group. And if you make animal models of Parkinson's using specific drugs such as NPTP, and then you give them GDNF, it seems that they can protect the cells and improve the function of these cells. And also, it's worth remembering that already GDNF has had several trials that's been performed, both from the UK, mainly from the Bristol group, but also in the US and other places. People have tried infusing this into various areas of the brain to see what happens in Parkinson's. And there, of course, the experience has been mixed. In the first set of trial, which was reported from here, it was reported that the GDNF is very well tolerated by patients who took very small number of patients. I can't remember the exact numbers. I think it's about 9. And in a few patients, these trials showed a very dramatic effect of reversal of the condition, improvement in several symptoms, the tremor, the bradykinase, when all other treatments were not working. However, then there was a large placebo-controlled study of GDNF, so where GDNF was mixed with a placebo and given in a blinded fashion to the patient, and the clinician also did not know, so so-called double-blind procedure, and that did not show the same degree of positivity. The second trial, therefore, did not show the same degree of positivity. Furthermore, there were some other concerns with GDNF safety. So as a result, the GDNF has not really led to the expectation that we had, but there's a trial currently ongoing again to address these issues. And it's also remembered that in these trials, the placebo arm, which is the dummy arm, also showed very impressive effects. So it's worth remembering that even infusion of placebo, which is inactive in Parkinson's, can actually produce some degree of dopamine, because dopamine stimulation is also triggered to some extent by expectation and reward type mechanisms in the brain. Now, just want to talk a little bit about Pdgf, Parkinson's platelet-derived growth factor, again another type of growth factor. Again, it's a protein that naturally occurs in the body. As I first said, protein, we're talking here about proteins that are naturally occurring within the brain. They again stimulate the cells to carry out various key functions, and one of them is about growth and differentiation of cells. And for instance, interestingly, the platelet-derived growth factor is already used for other therapies in skin repair, also for bone repair by our orthopedic colleagues. And the Pdgf-BB is specially formulated, especially being modulated so that it can be directly infused into the brain using a special delivery system so that there is no obvious toxicity. So it's again adapted for use within the brain in Parkinson's. So how does it work? It acts on the stem cells and the progenitor cells. Now, stem cells and progenitor cells, I'll quickly touch upon them in the end. They're basically building blocks. So all the nerve cells we have come from a generic, almost like a paternal body, the father and the mother, so to speak, of the cells. And these are the new nerve brain cells. So these are the progenitor cells, and the Pdgf helps on producing some specific cell lines from there. And it also helps these to multiply. And then the multiplication are close to the area where the dopamine nerve fibers are located, As a result, you would expect to see dopamine nerve formation and chemical formation secondary to infusion of the Pdgf-BB in the brain. And there has already been some studies performed in Parkinson's using this. In animal model studies, largely from Sweden, have shown that if you infused Pdgf-BB into the brains of animal models which have been made Parkinsonian artificially by drugs, it improves motor function, which is another key function of dopamine, if you remember. And secondly, it improves the dopamine deficits. All the symptoms of deficits of dopamine, the slowness, the difficulty in turning those animals experience, the speed of movement, all of these improve. And it is a long-lasting effect. One infusion has an effect for up to 3 months, another key difference from other therapies that we have at the moment. And there was also a clinical study in people with Parkinson's disease. So this is a phase 1, phase 2 safety and tolerability and efficacy study. Again, it showed a good safety profile, well tolerated. There was no obvious bad side effects. No differences regarding symptoms, however, because worth noting, just like the GDNF study, here the placebo arm also showed improvement. But another very interesting thing, if you look at a biomarker of the dopamine system, now what is a biomarker? For most conditions that we have, for instance, we have tests that you can do to confirm your diagnosis. In Parkinson's, we can do a DAT scan. Some of you probably had that. So that measures dopamine in the brain. And by using that, you can measure indirectly the dopamine content of the brain. And in fact, infusion of this showed in these patients that your DAT uptick improved after the Pdgf was infused. Now, so basically what happens is that the copy of the gene is inserted into the patient's body, in this case the brain. That will then instruct the cells to produce the protein. So that then will go on to produce, let's say, something like GDNF or Pdgf and so on. How can that be done? Well, already there's been some work done in this area. Well, in fact, quite a lot of work. It produces, you can either put it directly into the nerves. It can change the way the nerve cells work, and again, has shown effect on progression of Parkinson's. So in fact, the gene can drive a cell that's not working well to produce dopamine, instruct the cell to alter its way of working and produce dopamine in a proper way. So it can actually alter the signal to the cell. But the important thing is that to do that, you must insert the gene or genes into the right area of the brain. How can we do that? Well, usually we need to put the gene on a motor car or some sort of transport that will then take it to that part of the brain. And that can be delivered by a surgical process. And again, Alex would deliver that to a certain part of the brain. The bit that the gene is put on is called a vector. And in many cases, these are certain types of viruses that are used. And that suddenly causes, virus, my God, you're inserting a virus. Well, these are viruses that have been made sterile, so they're not infective anymore. Adenovirus is one of the examples, for instance. So the way it will work is you have a virus, you implant the virus with a particular gene, and you know that gene is going to work on your proteins that you want to produce. You then have that vector being implanted by a surgical procedure to part of the brain where that gene will then start working or instruct the deficient or dysfunctional nerve cell to produce dopamine. So that's the theory behind this strategy. You can actually have 3 main types of gene therapy. One group will produce growth factors, as we've already discussed. Another one can produce proteins, specific proteins directed towards just dopamine production. Another group will look at nerve cell activity, so they will just basically improve the activity of the nerve cells. So there's 3 different ways these gene therapies might work in the field of Parkinson's. And where are we with gene therapy? It sounds very exciting, but is it something that we can actually do in clinical practice? Millions and millions and millions of dollars and pounds have been spent on gene therapy research so far. However, I'm afraid that it's not really available for treatment option at the moment. For reasons and I'll talk about the reasons in a minute. It's still in clinical research. There are some trials that are currently underway, and initial results are showing some promise, but it's not a staggering, fantastic benefit that you would expect, which can't even bring in the possibility of a cure. And another big problem with gene therapy is safety, because you're in essence introducing an instructor to the brain, which is then instructing cells to behave in a certain way. Inherent to that are some risks, because are you able to control the instructions that are coming or that you're inserting into the brain? And that remains one of the fundamental concerns of gene therapy at the moment. And there is one drug that's already been tried, the Nurturin or Seri-20. This works through the protein pathway that I saw, GDNF. So in essence, it's going to be inserted into the brain, then instruct the cells to produce GDNF. It has shown positive effects in many animal models. And again, there is a slight problem that you might have to think about, is the disconnect somewhat between animal models and humans. The problem is, I don't think we still have a very good animal model for Parkinson's. As a result, many of these studies which have shown positive effects in animal model, clinically, however, has not shown that much of a great effect. This particular study showed that the gene therapy was performed on 1 or more regions. It was well tolerated, so there was no issues with safety as such, at least in the up to 1.5 years follow-up after the plants. But a recent study, which was reported by the company which developed this technique, was negative. So it did not show a positive effect, as you would expect, by measures that we do for these things. And these patients are still being followed up, though, because there is a chance with this sort of treatment you might get more longer-term benefit than a short-term benefit. So that needs to be seen. And then the final bit is cells. So we've talked about proteins, we talked about genes, and then we come to cells. So it's a sort of natural progression, really. So first we look at the genes that promote the factors, then we look at the factors, then we look at the cells itself. Can we do anything with the cells? Well, of course, you'll be aware, I think this is an area that's been extensively worked on since the 80s, really. In Birmingham, first cell transplantation was done in the mid-80s, I think, and then cell transplantation became fashionable in the late 80s with Mexican trials. More recently, there's been a lot of work on this area in the 90s, mainly from Sweden, but also from the US. And where are we? So they're stem cells you'd know a lot about, I think. And I'm often asked this question whenever I have a newly diagnosed case, person with Parkinson's in the clinic. They always ask me about stem cells. And essentially, what do stem cells do? Well, they replace brain cells and again, as you would expect, they try and produce healthy dopamine-producing cells. So just replacing the diseased cells by new cells. And that's the same principle people are now using for stem cell therapy in other conditions, such as, I don't know whether you recently saw some promise in stroke, for instance, in other sort of spinal injury, and so on. And these cells are grown from stem cells in the laboratory. So they're slightly different to other types of cell transplant I'll come to in a minute. So the advantage of stem cell is you're not dependent on any tissue donation, for instance. You can develop it from your laboratory. The other way, which has been used for a long time now, in fact, was the beginning of the transplant science, are fetal implantation. So these are aborted fetus, and you dissect out, these are very early abortions, you dissect out the dopamine cell from the brain, and that is then transplanted into the brain on both sides of a person with Parkinson's. And then these cells, then what happens, these fetal cells will slowly grow, they make connection with the existing brain cells in the brain, and then form almost a near normal cells architecture. And the great hope was that this is almost as close to a cure you might come in Parkinson's as it's possible. Of course, fetal cells have been implanted now since as I said, since early 90s. And what has been seen that these nerve cells survive up to the decade, or in fact longer. People have followed these patients up and done scans, which still shows, because by scanning you can show the activity of dopamine in the brain. But the improvement in terms of results are very, very variable. Some studies had shown some dramatic effects on some patients. I know of a patient in Sweden who is about 20 years on after transplant, who still don't require any significant treatment of Parkinson's. On the other hand, in other cases, we have not seen such benefits, and 2 large studies from the US, which did a placebo-controlled study, showed no benefit of stem cells, the fetal transplants over surgery. As a result, there is a big uncertainty. Does this really work? Is it really safe? Another safety is an issue. And there is also, of course, the big ethical issue about abortion and fetal cells. So those are the real problems when it comes to cell transplantation, particularly fetal cell transplantation in PD. So the stem cell therapy, if we go back to the stem cells now, research is still going over, and there's lots of units doing research, still really not ready for clinical trials. And the problems are the controlling the growth. In principle, there is still a worry that some of these cells, stem cells that are going into the brain, might actually turn into cancerous cells. There's nothing really to protect us from that at the moment. And when we crack that, then one can say that stem cell therapy may actually be the closest you can have to potential cure in Parkinson's. And just to go over there for the stem cell issue again, stem cells, remember, are cells that are being made in the laboratory, so we are independent of the ethical issues with fetal cells and so on. They divide and they can develop into different types of cells. So you can instruct a stem cell to go and form into dopamine cells. But that is where the problem lies, because at the moment, you can have stem cells, but the instruction for it to go on to develop into a specific dopamine cells is not very robust and strong. And again, we also know in Parkinson's is much more than just a dopamine problem. So when we can make these stem cells grow into the truly precursor cells, then I think we will be closer to the treatment strategies. In terms of where we can get them from, you can get them from embryos, either from the human embryo, or you can get them also from animals, models. And there's a lot of breakthrough that's being hopefully happening in terms of development of the stem cells. And in adults, it is not really possible to, if you have an adult stem cell, to a cell that's suitable for Parkinson's therapy. So if you already have a stem cell that's already grown, you can't really modify it. So what we really need to focus on is the embryonic stem cell, which is right at the beginning of its development. I have a diagram here. I'll very quickly go through it. I'm sorry it doesn't project, but essentially it's saying 2 types of stem cells here that we talked about. Here we have the embryonic form, And here we have the fetal brain. So this is the more desirable one. Essentially, we use a product called a neural induction, so the stem cells are then induced or instructed to go on through different pathways to finally become what we call dopamine cells. Yeah? So these are the dopamine neuroblasts, which then differentiate into dopamine, and this is what is injected into the brain. So you inject, once the stem cell induce dopamine neuroblasts, inject into the brain with the hope that this will regenerate some of the deficient function of Parkinson's in the brain. I've already touched upon this, but just very quickly, what are the issues, problems? Human embryonic stem cells are collected from embryos left over from IVF, and therefore immediately it raises a lot of ethical issues, particularly with the pro-life campaigners and so on. And there is the potential risk of uncontrolled growth. In essence, that can translate to almost a cancerous issue, and we do not have control over that at the moment. If you look at Ips cells, which is the pluripotent cells made from the laboratory source, that's of course of major interest because it removes all those ethical issues. They can turn into other types of cells, and you can be able to reprogram these cells. But again, the problem is that we haven't been able to really nail down how these cells can be directly directed to produce dopamine cells yet. We know that they're capable of it. Some evidence is gathering from animal models that it might be a case, but whether that can be translated to human is not known. And finally, if you use viruses to import new genes, again, there is the cancerous risk. So these two issues limit currently the role of stem cell therapy in the actual treatment of Parkinson's.
Thanks, Ray. Yes, I would stress I'm giving a personal viewpoint here. I'm not representing Parkinson's UK necessarily or anybody else. If I tell you a little bit of my background, I was diagnosed 4 years ago. The early symptoms I experienced, probably in common with many others, was a tremor. I found it increasingly difficult to use a keyboard on a typewriter because my fingers wouldn't do what I wanted them to do. I lost my sense of smell 10 years ago, something like that. I used to do quite a lot of speaking at conferences as well. And once when I was presenting at a conference, I suddenly found I was shaking, and I thought, you know, what on earth is wrong with me? And the sort of environment I was presenting to, people wouldn't be tolerant. They would regard that as that I was unconfident and not sure of what I was presenting. So it's much easier to present to a group like this where people will not worry if I shake or tremor. And because I know that you won't worry about it, I don't do it. So that's one of the peculiarities of the brain. When I was first diagnosed, I took an interest in research. I was fortunate enough to go to the Worldwide Parkinson's Conference 3 years ago, which was in Glasgow, which was a great introduction to all the research that's going on around the world. And I found that being involved with the subject, with the illness, actually helps me enormously. I spend a lot of time, not necessarily coming to conferences like this, but speaking, going to meetings, that sort of thing. I do a certain amount of work for Parkinson's UK. I'm actively involved in the local branch. I'm secretary of the West Hertfordshire branch. And locally we do a lot. We've introduced things like dance classes, singing classes, conductive education. So if any of you are from Watford or St Albans or Hemel Hempstead and you've got Parkinson's, you can find something different to do every day of the week. Because we're a very active branch. Anyway, I'm getting off the subject there, but that's a little bit about my background. So if we move on. That's it. So why take part in clinical trials? To some extent, it's obvious the objective is to find better treatments and a cure, ultimately, hopefully. It's obvious without trials there wouldn't be any new drugs. And I believe as well, as it says here, people with Parkinson's have a duty to help. We need to put ourselves out to some extent in terms of supporting clinical trials. I was talking to one colleague who was considering going on a trial. He said, oh, I would have had to stay in hospital overnight. I didn't want to do that, so I didn't bother to take part. And I think I wouldn't be critical of anybody, but I think that's perhaps not the best attitude to take. We've got to give up something to commit ourselves to taking part in research, and it's not necessarily easy to work out exactly what's needed. And the crucial thing is one of the main reasons that clinical trials fail is because they can't find enough participants. That's something that Ray touched on. I've seen several presentations on that subject, and that's a crucial thing, because people with Parkinson's often do want to take part in trials. People who are running the trials want to find participants so that they can carry out the trial effectively, but often the two don't meet. If you can't get enough subjects to take part in research, then the research falls apart. So it's very important to take part in research, and I think people with Parkinson's have a duty almost to help, and hence the Lord Kitchener poster. What do you get out of it? Obviously, a feeling that you are helping and contributing to finding a cure or a solution to a particular problem. I've said this, know the enemy, find out as much as you can about Parkinson's. That is something that has helped me enormously over the last few years to study and try and understand the condition, the latest research that's going on and that sort of thing. And by taking part in research, you get a much closer interaction with the research team, and you can ask questions more directly than you can if you visit your consultant. Consultant I used to say, I was in and out within 5 minutes. How's the medication going? All right, we'll increase it, we'll leave it the same, and that was it. If you take part in a clinical trial, the researchers are genuinely grateful that you're helping them, and they will spend time discussing what you're doing, what the test is about, and you will answer other questions. So hence I say you are someone. Another benefit is that your condition can be monitored more closely. If it's a continuing project, a tracking study or something of that sort, then you go for regular assessments and you can see how your condition is progressing. You get a better understanding. Obviously, the potential to benefit from new treatments, some of the drugs that have been mentioned by Ray earlier, and I've said can be fun, perhaps not often, but in Hertfordshire, the University of Hertfordshire has a dance research laboratory, and several of our members have taken part in experiments there, which they fill in questionnaires before and after taking part in dance routines, which everybody thoroughly enjoyed. So if you can find something like that, then that is an enjoyable, perhaps not clinical trial, maybe it's the wrong definition, but it's a trial anyway of a certain type of treatment. So what it means to take part in clinical trials, first thing is there are a wide variety of trials that you might take part in. I think people immediately think of a trial involving some new breakthrough drug that's going to provide a cure or a major impact. Some of the things, again, that Ray has described. But in fact, there are a wide variety of trials that take part. Sometimes they are to improve existing treatments. Could be a number of different things, different therapies like the dance sessions that I mentioned before. So a trial is not just a new blockbuster type of drug. What actually is involved, again, depends on exactly what the trial is, but typically there will be initially an assessment visit. You go meet the researchers, they ask questions, collect all your background information, and decide whether or confirm that you're eligible for the study that they're carrying out. Sometimes the study looks for older people, sometimes younger people. Length of time since diagnosis is also important, and it's particularly, a lot of experiments are interested in capturing people shortly after or within 2 or 3 years of their diagnosis. It's an interesting group. Explanations and consents. Again, Ray mentioned ethics committees and so on. There are ethics that govern the research that's carried out, and the researchers will explain everything that's required of you, will answer any questions that you have, and will get you to fill in necessary consent forms. And as I say, don't be afraid to ask any questions if you are taking part in a trial. The GP will also be informed if it's a drug-based trial, so that they know that you're taking part in something like that. Regular visits to monitor progress, again, if it's an ongoing study of some sort where you're taking a drug over a period of time or you've had a different type of treatment. I've said you don't have to leave home. There are now trials that you can take part in occasionally over the internet, filling in questionnaires and so on. And I'm going to talk about another trial that you can do from home as well. I said you don't even have to have Parkinson's. A lot of the trials look for sort of control groups, people who don't have Parkinson's that they can compare, and carers often take part in studies of that sort. There are all sorts of different things you can take part in. A word on test and control groups. If people can't read the caption, the mice in the front cage are sort of leaping about and going all over the place. The mice in the back cage are sitting down calmly, looking at them and saying, well, I guess we're the control group. Sometimes people say, well, there's no point taking part in research if I'm not going to be in the test group. If I'm in the control group, just getting a placebo, then there's no benefit to me at all. I'm only interested if I can actually get the treatment. That's not really the way to look at it. If you are allocated the placebo, well, you won't know whether you've got the placebo or not, but you still benefit from the other factors that I mentioned previously about being able to visit, talk to researchers, have your condition monitored, and you feel like an important person, an important part of the research. And it is very important to run experimental tests in that way, otherwise you can't conclude whether something has worked or not. Though taking part in test or control groups, it shouldn't put you off if you finally end up in a control group. So what is the role of the person with Parkinson's? Well, the obvious role is taking part in research. And I've said ideally as a partner, and there's a strong move to, as Ray mentioned, to involve people with Parkinson's in the research process. This is coming through and at the highest level from government papers that are being issued. I'll read out this quote in case people can't read it. It's from Sally Davies, who's the Chief Medical Officer for England. It says, no matter how complicated the research or how brilliant the researcher, patients and the public always offer unique, invaluable insights. Their advice when designing, implementing and evaluating research invariably makes studies more effective, more credible and often more cost efficient as well. And there's a strong move to involve people with Parkinson's in projects at an early stage to provide advice, and obviously they offer a unique perspective if they actually have the condition, that they can help to guide the researchers and the questions that are answered. So the role of the person with Parkinson's increasingly, and I think it's a very good thing, is becoming an integral part of the research at all stages. Now what happens after the study? I was given 4 things to talk about. I wasn't quite sure what to say what happens after the study. Again, if you can't read this cartoon, the 2 guys are sort of joined by a wire that connects their heads together. And so it says, so for the next 3 months, while your brain is healing, one of our neurology volunteers, Carl, will be helping you to think. Doesn't quite work like that, but you would be told at the start of the project exactly what's involved, when the project will come to an end, what your ongoing obligations are. So what happens after the study, it depends what the study is and what the parameters are. It may be a one-off study that you attend for a day. It may be something that you're tracked every 6 months, or other combinations. How to take part. This is very important. And my initial experience of this is I was diagnosed 4 years ago. I hadn't had a day's serious illness throughout my entire life. It was a total shock. And after partially getting over the initial shock, my feeling was, well, is there something I can do? Can I take part in research? Because I must be very interesting as a research subject. I'm not taking any other drugs. I'm prepared to volunteer for whatever people might want me to do. So I asked my consultant, and she said, well, I'll ask around. And I reminded her the next time I saw her, and she said, yes, I must talk to someone. But I never got any feedback. It didn't lead anywhere. I visited the Parkinson'S UK website, and I found there, those of you who work in the health service might know that there's something called Dendron, something called the Dendron list, which was a long list of research studies, half of which I think had finished, half of which were not recruiting anymore, and a small number might have been relevant, but it was very difficult to sort out and find the information as to what was going on. I found one trial I could apply for. There was an email contact address. I emailed and didn't hear anymore. So it was quite an off-putting experience. But this was 4 years ago, and I've said today things are much better. So how to take part? There are, again, several different routes to taking part in research. I'd highlight the Parkinson's UK and the Cure Parkinson's Trust websites, both of which contain a lot of information about research projects that are taking place and in an accessible form. I've put in the URLs there. You probably can't read them. I hope you'll get the charts or this information at the end. Obviously, online is the best way to obtain information. Also available something called the Fox Trial Finder. People will probably be aware of Michael J. Fox, the American actor who had Parkinson's, set up the Fox Foundation. They've introduced this tool which has been launched in the UK. Obviously, it started in the US, but you enter details about yourself, and it then produces a list of trials that you may be eligible for that you can apply for with contact details provided. So that's a very useful tool. Something that is intended to do the same thing. It's like these comparison engines that you use when you're looking for car insurance or home insurance. There are several available, and they look at things in slightly different ways. So NIHR, National Institute for Health Research Clinical Trials Gateway, is a UK-developed, well, it's a gateway, again, to enter information about yourself, your condition, and to try and match you with trials. It's not specific to Parkinson's, but Parkinson's is covered. I've said you can still try your consultant, and this little OK to ask symbol. There was a campaign a month or so ago encouraging people to ask their consultants about research they might take part in, and then you could go back to the website and enter what the response was. So within the health service, there's a move to, because I think consultants are crucial in this because it's the contact that everybody has. So I would urge all of you, next time you see your consultant, say, are there any research projects I might take part in? And ask them how to find out if you want to, because they will know particularly what local situation, local conditions are. So it could be valuable in that respect. And hopefully I'll get a better response than I did 4 years ago. A couple of other things. Parkinson's UK runs something called the Research Support Network, the objective of which, quoting that, to bring together people driven to help find a cure and better treatments. It's a group of within Parkinson's UK. You don't have to be a member of Parkinson's UK necessarily, but it's just a group of people who are interested in research. I think there are 500 or 600 members of that group now, and receiving regular information and updates about research opportunities and other things of that sort. It's a way to tap into information about what's going on in research terms. And those of you who are members of local branches can, what Parkinson's UK want to do is to encourage people to take the lead on research, report back to their local branches if they have a particular interest in the subject. Another thing that's available is visits to research projects. I've been on several visits, and of course we're in Oxford. Oxford University carries out a number of research projects for Parkinson's UK. I've been to Cambridge as well and London, so in this part of the world we're well placed to visit everything almost everything that's going on. And these visits are, I found, again, very beneficial and educational. I've been really impressed with the calibre, not only of the professors who are working on this, but also their students and their postdocs and people studying for PhDs and so on, and the level of interest and enthusiasm that the students show. And they're really pleased to meet people with Parkinson's and to show them what's going on. And when you've got people working in a lab with mice most of the time, it sort of provides a link as to what the end result or the outcome is of what they're doing, which is very important. Cure Parkinson's Trust has a website something called the Parkinson's Movement, which is described as being created by people with Parkinson's for people with Parkinson's. That takes a slightly different view. It's an international perspective. Again, it has news about the latest research, how to get involved, patient meetings, an excellent series of webinars that they've sponsored as well that you can access through the site. Patient meetings similar to this, I guess, and newsletter as well. I'll say a few words about trials that I have and am participating in. I'm sort of smiling slightly. One is TransEuro, which is actually a stem cell trial. We've heard comments about stem cells this morning. that is it's a large tracking study. It's funded by the European Union, being carried out in Cambridge, UCL in London, and also sites in Paris and Sweden and Freiburg in Germany, I think. And the objective there is to identify people who will go through to a stem cell implant. Those operations will take place later this year, perhaps in a few months' time. And again, people will be allocated to either the test or the control group. And I know I'm going forward with this, and I'm going to be in either the test or the control group for the first batch of people going through this. And I'm not put off even by, the possible. There are certain risks associated with it, but I feel it's something I should do. 23andMe, if people have heard of this, this is a very American initiative. The guy, one of the co-founders of Google, has his wife owns a genetic testing company. He found that he had some of the genes associated with Parkinson's. Sergey Brin, his name is, I was trying to remember it. And he's invested a lot of money in Parkinson's research. So you can actually have your genes tested at a remote distance. It's free for people with Parkinson's. All you have to do is apply through their website. They send material through the post. You spit into a tube and then post it back to them in California, and you get your genetic profile. The most difficult thing I had was returning the envelope through DHL, where you would have thought it was radioactive material in it, the way they went through all the forms and paperwork, because it's, could it be infectious in some way? But anyway, that worked reasonably, that worked well. I found I don't have any of the genes associated with Parkinson's. So it was interesting. The point about that research is their objective is to build up a large database of people with Parkinson's, to have all their genetics. You fill in lots of questionnaires as well, and they then correlate all that information to come to conclusions about different types of Parkinson's and people have. Another, again, this was some years ago, I've forgotten exactly what this one was about, but I went down to University College London for a day to have various tests and blood sample. But that was one I found early on. I can't remember what it was about, but I'm sure it was useful to UCL. I mentioned Parkinson's UK and Cure Parkinson's Trust. Hannah is here from Parkinson's UK. I think Helen is here from Cure Parkinson's Trust. So if you want to follow up with either of them, then please do. Now, I wanted to find, I think I'm at the end now, a quotation to finish with, and it was quite difficult. So I found something that, I thought was quite appropriate. You may or may not. One is a quotation from Martin Luther King Jr., which is, "We may have all come on different ships, but we're in the same boat now." I think that's quite appropriate for people with Parkinson's. Everybody has their own unique journey with Parkinson's, but in some extent, we're in the same boat in that we all have, or those of us who have Parkinson's here, a similar condition and would like not to have it and would like to find a way of finding a treatment or a cure. The other one's a quotation from Henry Ford. "Coming together is a beginning, keeping together is progress, working together is success." And the point of that is it's the point I made about working in partnership with researchers and people taking part in research, involvement at all stages. And it's by working together that hopefully we will find better treatments and hopefully ultimately a cure.
So the journey of, when I talk about drugs, I'm also talking about things that we inject into the brain and drugs in general. So this really encompasses everything. But the journey from the lab, where a molecule or a drug is first discovered or made, to actually becoming an approved medicine is a very, very long process. And these figures on here are not untypical. In general, preclinical tests, so that's lab type of testing, where either people are designing, these days many molecules are designed in the lab by researchers, by chemists, etc. Sometimes existing drugs or molecules are taken and tested in a different situation. So that part is called the discovery phase, or the preclinical phase. So you might be talking thousands and thousands of different products in the preclinical tests. And this period of the lab-based research is generally about 5 to 8 years. So this is before we as clinicians even hear about most of these things. If you keep an eye on the scientific literature, you might see some of these things. The majority of these molecules and various substances never even reach that phase, so they're tested in the lab and ditched because there's no promise. And then you have the clinical phase, which a clinical trial. So this is when a molecule or a drug is thought to potentially might work. So it's been tested usually in animals or in other models or computer-based models, but it has to be tested in people before it can actually become an approved medicine. And this would generally be either one drug or a number of very similar drugs. And this period takes about 6 to 10 years. So this is a very long period. And there are a number of phases of this. So phase 1 trials are really the safety studies. I'll talk about those in a minute. And those would be a small number of people. And that's really looking for toxicity of the drug. So it's looking at whether that drug has adverse effects rather than whether it works in that condition. Phase 2 would be a larger number of patients looking at not only a better safety study, but often an efficacy study as well. So in other words, how well does that drug work on the disease? So looking perhaps at symptom control or how it affects a particular aspect of the disease. And the phase 2 studies often involve what we call a dose escalation. So you can go up gradually and look at the toxicity of the drug. And also, as you go up in dose, does it work better? Then when you think that you've got everything right, you then do a phase 3 study, which would be a much larger number of people. These figures are typical for drug studies, so taking pills or other types of drugs. For the studies that we're talking about, that Ray talked about earlier, actually these numbers are much smaller, partly because of the expense of an invasive treatment, and partly because an invasive treatment is always going to be acceptable to fewer people. And then at the end of that period, there's a period of review. So the MHRA, the Medicines Health Regulatory Authority, for example, in the UK, and the FDA in the US, have to approve a medicine, and that period would typically be a couple of years, sometimes a bit more. So they'll take all the evidence that's there and they'll decide whether that drug can be approved for a particular disease. And these are the things that clinical trials are trying to find out, apart from the effectiveness that I've mentioned. So obviously effectiveness is very important, but they also have to look at safety and side effects of the drugs. And sometimes some of those safety aspects and side effects are only really known about when we do the larger studies. Because if you have, say, a side effect that only occurs in 1 in 1,000 people, and you've only tested 500 people, you might miss it. And in fact, there are phase 4 studies which go beyond the initial regulatory approval, looking at very large numbers of patients that sometimes pick these things up. And there are a few things every so often that hit the headlines. For example, I think the Vioxx for inflammatory pain, which was found recently to have effects on the heart, and that was really after it had been released 10 years ago. So there had been many, many thousands, probably tens of thousands of people who had taken it. So the other aspect that a clinical trial is trying to work out is what is the best dose, because when we go from rat or primate to human, humans are very different. And so the doses that are worked out in the lab are not always the same as the best doses in the human. And so the clinical trials have to be designed with that in mind to try and have some variation in the drug dose. So phase 1, 2, and 3. This actually is not really. This is slightly misleading because there are phase 1, 2, and 3 trials are actually in that middle phase after the lab. So they're the clinical phase. So just to reiterate then, phase 1, typical numbers, generally quite small. In your average drug study, this would be healthy volunteers. So these would be people who are happy to take doses of the drug but don't have the disease, just to look at the safety profile, and to look at side effects of the drug and what we call the pharmacokinetics, which is what happens to that drug when it's in the body. Now, the trials that we're talking about with invasive treatments in the brain, this phase 1 is not likely to be healthy volunteers, because we'd probably have difficulty getting a healthy volunteer to agree to have a tube put in their brain and an infusion of a substance. I guess if we paid them enough, they might be prepared to take part. So this phase 1 may actually be patients with Parkinson's disease who are keen to see whether that drug is safe and might be used. The phase 2 is typically hundreds, but again, with these invasive treatments, the phase 2 trial, typically with the ones that Ray was talking about, is typically about 30 or 40 patients, maybe 50, 60. So it's less than fewer patients than the drug trials, for example. And this phase is to look at effectiveness. So this is getting data, getting people to take the drug, usually blinded, so that they don't know whether they're getting the active drug or not, and looking at the two groups and proving statistically that the group who are getting the active drug are doing better. Then the phase 3, so this is pre-market, this is before the drug can actually be marketed, large-scale trials with larger numbers of people to look more on the effectiveness in a larger cohort of people, but also looking for those safety side effects that might be less common, for example. And because you've got more people, you can look at more other factors such as what drugs people are taking, etc. So these are the typical numbers we're talking about with protein, gene, or cell therapies, 5 to 20 in phase 1. In fact, the Pdgf trial, I think, had a small, I think it was 10, and was about 10 patients. The GDNF trial had 9. Some of those viral vector therapies had about 10 to 15. And again, phase 2, slightly smaller numbers than you'd expect with a drug therapy. And then depending on the side effect profile and the efficacy profile, this number in the phase 3 is really depends on those initial studies. Let me just give you a little bit about some of the jargon that we've talked about. So placebo is a treatment, it's not a treatment, but it's something that's administered to make you think you might be getting the treatment. So it's, for example, if you're giving an injection, instead of injecting the drug, you could be just injecting water. So that would be a placebo treatment, or a pill with no active ingredient. And the point of having that is that we can determine that the active treatment is better than not having the active treatment. Because there might be an effect of actually coming to the clinic, being seen more regularly. There might be a psychological effect of having the treatment, which is not actually related to the actual drug. So in the placebo arm, the people in the placebo arm are getting all of that as well. The only difference is the active drug. Double blind just means that neither the patient nor the clinician who's running the trial or recruiting the patient knows whether that patient is on the drug or not. And that's important because that gets rid of bias. So if I put a patient into a clinical trial and they agree to go into that trial and we both think they're on the active drug, or we know, they might get a better response because of the placebo effect. Inclusion criteria is just certain criteria that mean you can go into that trial or not. So it may be that you have to have a particular type of Parkinson's symptoms or a particular stage of the disease. Exclusion criteria are things that stop you from going into that trial. If you've got a problem with your blood clotting, for example, you might not be allowed into a trial that involves surgery, or if you're pregnant, or if you're under 18, or something like that. DSMB is the Data Safety Monitoring Board. This is an independent board, so for every control trial that we have to have an independent set of people looking at the safety of that trial. So, for example, I've been asked to be on the data monitoring board of a trial for GDNF in Bristol, which I will look at the data, and if there are any safety issues, I would actually have the power to stop that trial, or as part of a committee, not me, obviously. So that's a very important independent monitoring board, and there are lay people, etc., on that. Randomization is what we do to decide who gets the placebo and who gets the active treatment. So there are various ways of doing this, but one way would be to have half of the patients who are having the treatment in all in an envelope where you can't see what's in it, and it will say yes or no, are you getting the treatment? Or you're just given the vial of drug and you don't know whether it's got it in. How much does it cost? Well, by the time a drug reaches phase 3 trial, nearly £1 billion generally will have been invested in the development. So if you imagine that it takes, if you look at the success rate, out of 5 to 10 potential medicines that go through clinical development, only one will receive regulatory approval. That's 10 to 20% after it's gone past the lab phase. So that's why it's such an expensive process. If you imagine the 10,000 molecules that are looked at in the lab, it adds even more money. So it's a very expensive process. Setting up clinical trials is expensive. A typical clinical trial could cost 50 to 80 million pounds at the phase 3 trial, sometimes more. The highest failure rate is at phase 2, i.e. the one where you've gone through the initial safety study, but you're looking at whether it works or not. That's about 82%. Once you get past that, you've got about a 50% failure rate. By the time you make it to phase 3, about 2/3 will fail due to lack of efficacy. So it's not a cut and dry thing. It's once you embark on a particular drug that you think might be good, there's still a high chance it won't get there. This is just sorry about the size of the print. It might be a bit difficult to see, but this is just looking at the time at different stages. So drug discovery, 4 to 5 years, preclinical testing for another 5, phase 1, 7, 8.5, 11. So it takes a long time to actually get there and grow into something that's useful. So what happens after the clinical trial phase has happened and you've had your phase 1, phase 2, phase 3 clinical trials, how does that drug actually get into the general population of people with that disease? Well, what happens is that you have a registration period where the findings from those clinical trials are submitted to the regulators, such as the MHRA. They carry out an assessment looking at the safety and whether the trials are really good quality trials that have been conducted well. And then if they're happy, they'll give licensing approval. And then once it's approved, that drug can then be marketed by the companies that are making it. And then I mentioned phase 4 trials, which are carried out after a drug has been given licensing approval. So these are even larger scale trials that are used to see whether a drug is working or not. And also long-term follow-up is important. So the industry has a commitment to long-term follow-up and looking at the 10-year, 20-year stage to see if there are problems related to that drug. And the industry is regulated by the ABPI in the UK, which oversees that process. Regulatory perspective is important because you have to have some sort of third party deciding whether industry and whether clinicians are doing the right thing. There are different medicines coming in different formulations. So, for example, you may have an active drug in a tablet, but it may be that in a certain condition, having it as a liquid or having it as a spray, nasal spray, might be more useful. But if you want to do that, you still have to get through the regulatory hurdles to get through to be able to do that. So you can't just change a drug or the way it's formulated to suit. It still has to go through that regulatory approval. So that's another important aspect. So most people, when they think about medicines, probably think about tablets, but actually there are over 50 possible ways of delivering drugs into the body. And we've talked today about injecting things into parts of the brain. That you can give things by injection into the bloodstream, into muscle, nasally, by liquid, through other body cavities, all sorts of things. So these all have to be tested. And what we're talking about here is drug delivery directly into the brain. And I think Ray's already talked about the blood-brain barrier, so I'm not going to talk about that again. But to get past the blood-brain barrier, you have to put things intracerebrally. So you have to put them either directly into the brain tissue or directly into the fluid spaces in the brain. And that means surgery. Intrathecal would be generally done via a lumbar puncture in the back. So it would be a needle into the back, into where the nervous system is, but without having to go through any nervous tissue. So that's always another possible way of doing it. We've talked a little bit about placebo. I don't think I'll mention that again. But just to say that placebo is very important because there are many trials that have fallen apart because there has been improvement in the placebo arm. And the key question that any trial has to answer is whether the active drug works. And there are many other factors. For example, the UK, the Pdgf trial, which was a UK trial for deep brain stimulation for Parkinson's disease, it showed that deep brain stimulation was effective compared to the placebo. But actually, if you look at the placebo arm, there was still quite a significant improvement in the Parkinson's disease symptoms of the patients in the placebo arm. It wasn't as high as with the deep brain stimulation arm. And that's a good example of how being part of a clinical trial, because you get better care perhaps, you get better follow-up, and perhaps in that trial people's drugs were better maintained and adjusted, you can actually improve in both arms. So placebo is very important, and this is a regulatory requirement. And this has to be done even in invasive therapies. And of course, when you're injecting things into the brain, this has all sorts of other challenges. So what is our top priority? Our top priority is always to ensure a trial participant's health is not at risk during a clinical trial. So for that, we have the Data Monitoring Committee that oversee what we do, but we also have our own adverse event reporting during a trial. So in any randomized controlled trial, whenever there is any side effect, whether it's due to the medication or not, it has to be reported. So we have what are called adverse events. So if a patient in the trial gets a rash, or they get headaches, or they collapse, that has to be reported in all groups. And even if we think it's not related to the drug, we still have to report it. So if a patient, for example, trips up a step and we don't think it's related, we still have to record it. We can record it as we don't think it's related, but it still has to be recorded. And that's important because that's how you find out about the rarer side effects that you might otherwise not notice. So surgical placebo trials are slightly different to drug placebo trials in that when we do surgery, generally it's not considered ethical to do surgery without some sort of benefit. There are trials that have been done with surgery where people have drilled holes into the skull under anaesthetic and not told the patient whether they're getting the drug or not, or whether they've got the treatment. And those have been considered to be ethical. But I think generally the way most things are going is that we discussed earlier about the open label treatment. So everyone gets the surgery, but if it's shown to be effective treatment, then at the end of the trial, the people who haven't got the treatment still get the chance to get it. And of course, what's important is that patients and their carers are informed about the possible risks, what are the advantages of taking part and what are the disadvantages, and what it will involve being part of that trial. So it's all about risk and benefit, and I think Richard has given us an excellent talk about the advantages of taking part in trials. Of course, there are risks, such as surgical procedure. There's a risk of a drug that you don't know whether it's going to necessarily work or whether it's going to have side effects. Obviously, that's been tested as much as possible before you get to that stage, both in the phase before you get into the trial, but also in the lab. But I think the benefits generally would outweigh the risks, and otherwise we wouldn't be doing it. You tend to receive very good medical treatment as part of a trial. Obviously, you should receive that treatment anyway, but it's at no extra cost. You do have the chance to get an experimental treatment that otherwise might take you another 10 years or so to get if you wait until it comes to the post-market phase. So there are advantages with taking part in trials. And we talked obviously about risks versus benefits.